In 2014, the global health community was tasked with preventing an outbreak of the Ebola virus, and despite the CDC's largest response to an outbreak, the virus was only recently contained. Education of dentists is essential because a major transmission mechanism for Ebola is through mucous membranes. Ebola RNA virus damages the immune system, causing bleeding inside the body, and can damage almost every organ. It first targets immune cells, such as macrophages, which engulf Ebola by macropinocytosis and clathrin-mediated endocytosis, where the virus is then able to replicate and be secreted. Apoptosis is activated in the infected cells, and breakdown of endothelial cells causes blood vessel clotting, leading to bleeding. Stopping endocytosis of Ebola will prevent infection. We compared whole genomes of individuals immune to Ebola infection to similar individuals who are not immune and found mutated clathrin receptor proteins. These individual cells do not recognize Ebola, thus preventing Ebola from entering the cell. Using CRISPR-Cas9, the mutated sequence can be custom sequenced, attached to a DNA cutting enzyme with the old clathrin gene being deleted and a new gene inserted. The cell would then express the mutated clathrin receptor protein that is resistant to Ebola. A target sequence of RNA will bind directly to the patient's human hematopoietic stem cells with CRISPR-Cas9, inserting DNA that codes for the mutated clathrin sequence. The engineered cells are then injected into the patient's bone marrow, where they directly edit other stem cells and newly formed leukocytes. The stem cell then produces progeny that also display the mutated form of the clathrin protein. Newly inoculated cells can then spread throughout the body, expressing the mutated clathrin receptor protein and eradicating the deadly Ebola virus.